Dear children, namaste and welcome to session 2 of our class 10 CBSE biology chapter, which is Heredity and Evolution. This is Ambika, your biology master teacher, right here on this amazing platform of Vedantu. Okay, guys, so let's get started. As you know, this chapter, I think I told you in the last session also that I would now actually like to call this chapter just heredity or genetics, basically because as we know, evolution and its theories have been removed from the syllabus. So anyway, let's go on and understand more about this very, very interesting topic. Okay, but before that, something positive, something motivational, and something inspiring for you. One small positive thought in the morning can change your whole day. Likewise, actually, the flip side of it, one small negative thought in the morning can also change your whole day for the worse, and this one for the better. So, try incorporating this in your daily lives, children. The moment you wake up, take a deep breath, take a few deep breaths, just stretch out completely, and tell yourself something very positive. It can be anything, as simple as you want it to be, okay? But definitely that energy will stay with you for the rest of the day. Okay, so let's move on and look at the homework question of the last class, and that's about sickle cell anemia. Remember, what variation in sickle cell anemia patients makes them resistant to malaria? And I told you to do your bit of research about that, right? So here is the answer to the question. Sickle cell disease is basically a blood disorder in which there is a single amino acid substitution in the hemoglobin protein of the red blood cell. So what exactly does it mean? I think I told you in the last class that there are amino acids. Remember, I told you about transcription, translation. I remember just vaguely mentioning it to you. So what happens is, it is because of the assembly of amino acids in the ribosomes that protein synthesis would occur. Hemoglobin is one such protein in our body. Hemoglobin, just like any other protein, is made up of chains of amino acids. So because of one simple mistake while making the amino acid chain during hemoglobin synthesis, this is what is a single amino acid substitution, wherein instead of one particular amino acid, by mistake, there is another amino acid which comes into place, okay? And because of that mistake in the hemoglobin protein of red blood cells, it causes these cells to assume a sickle shape. What do cells actually look like? What do red blood cells actually look like? Let me just draw it out for you so that you might understand it a little easier. We know that normal RBCs would be circular, right? This way. Whereas in the case of sickle cell anemia patients, the red blood cells assume a sickle shape, literally like a sickle. What exactly is a sickle? It's a tool which is used in agriculture. I know that's not the perfect shape, but roughly like this, okay? So obviously, what do you see? What's the problem with this? Obviously, the surface area of red blood cells is greatly reduced, and because of that, oxygen carrying capacity of those red blood cells is greatly diminished. Right, so this is a huge problem. Sickling and sickle cell disease also confers some resistance to malaria parasitization of RBCs, because actually it comes as an advantage to them sometimes, because malarial parasite, which is Plasmodium, is said to complete its life cycle in the red blood cells of humans, right? So in such patients, malarial parasitization of RBCs becomes a little challenging for the parasite. So this means it acts like an indirect resistance to these patients to malaria, so that individuals with sickle cell trait have a selective advantage in environments where malaria is present. So in places where malaria is prevalent, sickle cell anemia patients have a slightly better advantage as compared to the others, because some kind of resistance, extra resistance, is there for these people, okay? So in that context, it acts like a blessing in disguise, but definitely this is a major problem which can give such patients a lot of difficulty. So I've got the answers from Ayush, I think that's Ayush Yadav. And then we have Isabella. Both of them have given me the answer completely and at first. So thank you very much, Ayush and Isabella, and everyone else who managed to take off a few seconds or a few minutes to do your little bit of research and give me the answer to this. Thank you so much, Ayush, for also mentioning that you loved the session and the session was superb. Thank you so much, children. You know what, as teachers, these tiny tokens from you all would be absolutely enough to make our day completely. Okay, so thank you so much, children. So let me also share happy news with you. The JEE Advanced Results are out, as most of you know, and Vedantu students have bagged top ranks in IIT JEE, the exams, and these are said to be the best results amongst all online classes. So imagine in this year of pandemic 2020, although so many things in the world might have gone on halt, at least temporarily, especially in the field of education, where schooling and a lot of other things, a lot of training and other such things had to shut down, at least temporarily, Vedantu never stopped, especially the teachers, the students, the parents of such students. And that is why we are specially making it a point to celebrate all our super scorers. You can look at all of these wonderful faces of all of these children. These are some of our super scorers, and there are many such super scorers. So children, definitely take inspiration from them. In the sense, like, as I always tell you, I'm not saying that all of you must attempt JEE, because that's personal choice. Children who want to, children who are already JEE aspirants, certainly take direct inspiration from them. But otherwise, the rest of you also, if they can achieve this in a year like 2020, certainly you guys will also be able to do exceptionally well when it comes to your board exams. Trust me. Just have faith in yourself. Just do your part and the rest will follow. Okay, so today let's learn about Mendel and the pea plant, about the monohybrid cross, dihybrid cross, and the 3 laws of Mendel. Children, I really hope that you have come prepared for this session by being thorough with whatever we have discussed in the last session, because remember, all of those terminology I told you are very, very important. Today I won't have the time to explain each of those terms in detail, but rather I would be using them interchangeably. So in case there is any term that you find difficult to understand in today's session, it's basically because you're not thorough with what I told you in the previous session. So I would suggest go back to that video and watch it and then come back to this. Okay, so Gregor Johann Mendel, we call him the father of genetics, because in the 1800s, back in the 1800s, Mendel is said to have been a priest. In fact, he was a priest, and he lived in an Austrian monastery. He was an Austrian monk, and at the same time, he was a very, very passionate gardener, and he was also a great mathematician. So his curiosity in everything inspired him to grow a garden of his own, and he had a beautiful pea garden. Okay, and the plant, the main plant that he used to grow there was the garden pea plant, scientifically called Pisum sativum. Okay, so it was in fact from the pea garden that all of the theories of inheritance that we know about today started. The story of all of these laws came from there. Today we can say if the parents are both belonging to O blood group, the children would also belong to O blood group. If the parents are AB and AB, the children can be A, B, AB, O, anything, and so on. We say a lot of such things because of whatever Mendel came up with. And the best thing to be appreciated about Mendel is the fact that he discovered all this exclusively based on his keen sense of observation, because that was a time during which not even proper microscopes were not even very popular around his times. And also, the world knew nothing about chromosomes. The world knew nothing about anything, genes, alleles, nothing at all. Whatever I told you in the last class, I thought of telling you so that it would help you understand this better. But then Mendel, without having any of that background information, he went about with the help of his own instincts and coming up with a theory. So how did the story start. Mendel was just as with any other scientist, he was just very curious to understand how the theory of inheritance worked. So he went about trying to understand how to observe this, how to come up with theories of inheritance. That is how he finally came up with a plant, and that is Pisum sativum, the observations on which led him to come up with the theory. So, but why did he choose Pisum sativum? Why didn't he use human beings or any other animals as his type study organism? For the simple reason that if at all Mendel had to use, if at all Mendel had to observe human beings many generations together, of course, if he had to observe them, obviously one lifetime wouldn't be enough, because Mendel himself is a human being. So human beings out of question. Any other animal that he wanted to study, like rats and things like that, because he was a monk and there were certain restrictions at his monastery, like studying animal sex was a taboo and things like that, he decided to stay away from all that, and he went about into his humble tea garden, and that is where all of this started. So based on his observations, he came up with three theories, which we call the law of dominance, the law of segregation, and the law of independent assortment, about which I will tell you a little later today. Okay. He also found that gametes, we know what are gametes, right? Sperm and ova. He discovered that gametes have factors which are responsible for expression of character in offspring. What are these factors? Today we call these factors by a special name, and that is gene. In fact, for gene and allele, for both of them, Mendel used the term factor. Back then, the terms gene and allele weren't even discovered. Okay, so let's move on and understand more about why exactly Mendel selected the pea plant and not any other plant. Why it was a plant, I have told you, but why the pea plant itself? There were a few special reasons, and these are some of them. Pea plant has bisexual flowers because it's self-pollinated normally, but it can also be easily cross-pollinated. That's important, right, for a scientist, because whenever he has to carry out experiments according to his convenience, something that can be cross-pollinated does a lot of advantage, especially in genetic studies, right? Then, secondly, pea plants have many varieties which can be true bred. Okay, true bred, pure breeding, all of that I have told you in the last class. And annual plant which produces a large number of offspring. This is an annual plant which produces a large number of offspring. So it has a short lifespan basically, and within that short lifespan, every time it reproduces, it produces a large number of offspring. Why is that an advantage? For a scientist, when a huge sample is available for observation, especially in a field like genetics and inheritance, it's a huge advantage. Better and more accurate would his results be. And pea plants were easily available, they could be easily handled. That was another advantage of the pea plant. This is why he had a pea garden of his own. So yes, these are the major advantages of pea plant. So just in case you have forgotten what true bred or pure bred means, children, pure breeding or pure bred individuals are those which have been exactly expressing the same trait over many generations together. Okay, they have remained unchanged over many generations together. Okay, so you will understand better once we start talking about Mendel's experiments. So these were the seven major characters. These were the seven characters that Mendel observed in the pea plant, because he knew that in the pea plant there could be some pea plants which were tall, some of them were dwarf, some of them had an inflated pod shape, some of them had constricted pod shape, some of them had round seeds, some of them had wrinkled seeds, some of them had yellow seeds, some of them had green seeds. Some pea plants he saw that they had axial flowers, some of them had terminal flowers, some of them had purple flowers, some of them produced white flowers, and of course, some of them had a green pod color, and some of them had a yellow pod color, and so on. So, because these seven characters, in these seven characters, a pair of contrasting traits could be observed. By traits, I mean versions of characters. For example, in stem length, the versions are either tallness or dwarfness. These are the traits of the character's stem length. Likewise, for the character pod shape, the traits are inflated and constricted, and so on. So there are clear-cut contrasting pairs of traits observable for these characters. And this is why Mendel chose the pea plant, because it makes it easier for him to observe, right? Make his observations on the studies of genetics. So this was the main reason. And whether it's dominant or recessive, how is it that Mendel knew that? That's a different thing. This is actually, this table has been based on our understanding today. Whereas when Mendel started off, he did not know what was dominant, what was recessive, or anything like that. It was only at the end of his experiments that he understood what is dominant and what is recessive. Okay, so let's move on. Children, before I give you a very quick quiz, let me tell you something. Let me remind you about your golden opportunity to get closer to the KBC hot seat. We know that KBC 2020 is around the corner, and for the student special week, Vedantu students, sincere Vedantu students, have a golden opportunity. And how do you get closer to the hot seat? It's very, very simple. Just download Vedantu app right now from Google Play Store and start playing V-Quiz every day till the 18th of October. Okay, it happens at 11 a.m. and 7 p.m. every day. So just play V-Quiz every day till the 18th of October, 11 a.m. and 7 p.m. And your chances of getting shortlisted for the KBC hot seat would become a lot higher. Okay, so do not miss out on this golden opportunity. I remember when I was a child, when I was in school, I used to dream about some chance of meeting Mr. Amitabh Bachchan and going and contesting on KBC and all of that. I'm sure many people in my generation would have been like that, because even back then they used to have Children's Special Week and all of that. But anyway, we never got an opportunity. So your opportunity is right here. Do not miss out on that. Okay, so let's answer a few quick questions. Mendel chose the pea plant because it has unisexual flowers, it has a short lifespan, it produces a few offsprings in a single round of cross, or is it all of these? Which of them? And your time is up. The answer is it has a short lifespan, because A and C are actually wrong statements. The pea plant has bisexual flowers and it produces several offsprings, many offsprings in a single round of cross. Okay, considering that, it's only B which is correct. And your next question is here. In case of pod color, the color green is dash. Dominant, recessive, mostly recessive, but sometimes dominant, none of these. Not a problem in case you get this wrong, because it's a purely memory based question. So in case you remember the table which I showed you last, please try and give me the answer. And your time is up. The answer is dominant. Seed pod, green color of seed pod in a pea plant is said to be the dominant trait over yellow pod color. Now, round and wrinkled are contrasting traits of which of these characters? Shape of pod, shape of seed, color of seed, or flower color? Round and wrinkled. And your time is up. Absolutely, it is shape of seed. For this question, I wouldn't agree that it's a memory-based question, basically because even using your common sense, you know that if it has to be round, it has to be shape of seed. Seed pods cannot be round as far as a pea plant is concerned. Okay, so good job, guys, if you have got it correct. So let me tell you a few more useful things for you. In this year, during this period of online education, these are the most probable problems that most of you would be facing. Unsolved doubts, no proper notes because school is not happening regularly. Tests and assignments, the way in which they happen itself has changed. Competitive exam preparation, choice of schedule, choice of language, all of this is a problem. But then it's not a problem anymore, not to worry, because we at Vedantu have got all these problems solved for you, in addition to giving you a lot of bonuses. And what are they? Unlimited live classes, micro and crash courses, performance reports, personalized attention, and whatnot. I think newcomers, if at all you're here, you might wonder what micro courses are. So children, remember, micro courses are chapter level or topic level or concept level courses which have been designed especially for those of you who want to strengthen your grasp over one particular chapter or one particular concept alone. Okay, so crash courses, of course, you all would know, but we also have micro courses which many of our children find very, very useful. Okay, so What do you do to know more about this? Just visit the link given in the description box below and the pinned comment, and use the coupon code AMBPRO to avail the best offers that we have out there in terms of the amount. So how much does it cost is one question that most children ask, or most children at least have in mind. So I will explain everything to you in a transparent manner so that you don't leave the session with a doubt. So just click on the link in the description box below, children, and choose your grade. Okay, first of all, you have to choose your grade. So I'm assuming for you guys it's going to be class 10, and that too it's going to be CBSE 2021, or if it's anything else, choose it appropriately. Check out the details which are all mentioned here, the details of what we call Vedantu Pro. Most of them I've listed out to you already, but here are some more bonus features which you can just go through to get a better idea about, and click on the button that says get subscription. Okay, now what? Now this is where the question of payment comes. So I am explaining to you three major plans that we have: 1 month plan, a 3-month subscription plan, and a 6 months of subscription plan. So for a 1 month of subscription, what does it mean? If you subscribe to Vedantu Pro for 1 month, a 30-day subscription, this means that for that entire 30 days, you get unlimited access, you get unlimited live interactive classes, test series and analysis, assignments and notes, and all of those features which I have told you. That too, for all your major subjects together, your science, social studies, maths, everything covered. And for 1 month, normally you would be charged rupees 2,699. Okay, whereas currently, because of the offer that we have, apply the coupon code AMBPRO, okay, and get the amount payable reduced to 2,159.2. Okay, this is for a 1 month of subscription. For a 3-month subscription, which means for the next 3 months, approximately 95 days of subscription, for those 3 months, you will get unlimited access to all of these features. The normal pricing is 6,999, whereas application of the coupon code AMBPRO, the same code that I told you, would reduce your amount payable to 5,599.2. Okay, this is for 3 months. And finally, for 6 months subscription, which means for the rest of this academic year, basically, you can be tension-free and leave everything to Vedantu. You would normally be charged rupees 11,499. Whereas if you apply the coupon code AMBPRO, you get it available, you get it reduced to 9,199.2. Okay, well, personally, children, I think this is one of the most reasonably priced courses out there in the country, because as a parent myself, I know how expensive education these days is. The price is only going up each day, each month, each year. But at the same time, at Vedantu, there's a very strong passion for all the employees and everyone who works together, all the teachers and all the people who work even at the back end, that education has to be made affordable to all. So this is why, children, we have also transparently given you all the details. It's completely up to you now. Just give it a shot, because great quality content available at a reasonably priced rate is not a joke, and it's not something very common. So definitely give it a shot and make the best use of it. Okay, so let's move on into Mendel's experiments. So how did Mendel start? Let's consider one of those 7 characters that Mendel chose. I told you there were 7 characters, right? That Mendel, like when I started the session, I told you there were 7 characters which Mendel chose. For all those 7 characters, he performed experiments, but then let's do it only for one experiment, because the results for all the other characters were also the same for Mendel. So let's start with the character plant height or stem length. Okay, because Mendel knew that there were tall pea plants as well as short pea plants. So Mendel raised pure lines with the selected contrasting traits, by which I mean Mendel made sure he chose 2 parent plants, parent 1 and parent 2. Okay, these parents were similar in 6 characters, and the only character they differed in was plant height or stem length. One of them was tall and one of them was short. He just went about raising pure lines in them. In the sense, P1, he chose a tall plant as P1 by ensuring that this plant, this particular plant, had been having only tall plant ancestors. All its ancestors had only been tall. And this one, short pea plant, he chose such a plant that had only short pea plants for many generations together. So this took him some time to develop the parent plants also. He ensured that they were purebreds, and that's for a very important reason, which you will understand in some time. So one tall plant and one short plant were the parents. He crossed them, cross pollination, and he observed that all the offsprings were tall. Mendel was surprised. So, what happened? Doesn't the second parent, I would like to call this P2 and this one P1, so to make it easier for us to communicate. So, didn't P2 play any role at all? Wow, it's not even like an intermediate height. All the offspring are tall. How did that happen? So, he called this generation as the first filial generation. First filial generation. Okay, yeah, it's mentioned here already. F1 generation or the first filial generation. So Mendel was curious. Now do remember, one important thing to note here, children, is the fact that what is represented here does not mean that only one progeny was produced. Okay, do remember that. There might have been hundreds of them. There might have been like 50, 60 of them. Whatever the number was, he observed that all of them were tall. It's only the ratio which is mentioned here in this image. Okay, parents, of course, there are only two parents, obviously, but do remember, pea plant produces many offsprings in one go. So then what he did was he went about self-pollinating the F1 generation, self-fertilizing them. So the F1 plants were self-pollinated, and the resulting generation, which he called the F2 generation or the second filial generation, he observed them. Now, to Mendel's surprise, in the second filial generation, he saw that 75% of them were tall. Only 25% of them were short. Which means if you take the ratio 75% and 25% basic math, it's going to give you the ratio 3:1. 3 tall is to 1 short or 1 dwarf. Which means if at all there were 100 plants which were produced here, 75 of them were tall, 25 of them were short. So Mendel was like absolutely shocked. Okay, now in this cross, actually in the cross which happened now, it means that it was self-fertilization, which means the sperm and the gamete were all from this generation, the first filial generation. So despite having a sperm and ovum from a plant that has only been tall, how come a short plant cropped up? Did it magically inherit it from its grandparent? Or is there some other underlying mechanism to it? So Mendel was very curious to know, and this is the hypothesis he came up to. He assumed and he came up to the hypothesis that in an organism, there is a pair of factors that controls the appearance of a given characteristic. Okay, today we know that it is genes and alleles that we are talking about, that Mendel was talking about. An organism inherits these factors from its parents, one from each. There is a pair of factors, one of them from one parent, the other one from the other parent. A factor is transmitted from generation to generation as a discrete, unchanging unit. Okay, and when gametes are formed, the factors perhaps separate out and are distributed as units to each gamete. And if an organism, for example, has two unlike factors, which we call alleles, we've seen homozygous, heterozygous, and all of that. So Mendel says that if at all an organism has two unlike factors, okay, which means we are talking about the heterozygous case specifically, one may be expressed to the total exclusion of the other, the dominant versus recessive one. This is what Mendel came up to. So yes, with that understanding, right now let us work out with the allelic combinations also. So Mendel's purebred parent organisms had the genotype capital T and small t. So this is why I said purebred. Having purebreds is very important so that they are not heterozygotes. Okay, pure for tall, which means homozygous tall. Purebred for dwarf is homozygous dwarf. Okay, obviously the recessive one would always be homozygous anyway. The gametes they produce obviously would have just one copy of the genes, one copy of the pair of alleles that were there in the parent. Because we know gametes are haploid and the organism, on the contrary, is diploid. So this is the type of gametes that this parent produces, and this is the type of gamete that this parent produces. When they both come together during fertilization, F1 generation has the genotype capital T small t, which is a heterozygote. Okay. Now in this case, when it is selfed, what happens? The gametes that a heterozygote would produce would be of two types. Half of the gametes would contain a capital T, while the other half would contain a small t. Likewise, because it is self-pollination, the other parent also has exactly the same, because it's basically selfing. Okay, so these are the gametic combinations, and work out all the possible combinations. This and this together gives you a homozygous tall plant. This and this together would give you a heterozygous tall plant. Another combination would give you again a heterozygote, and this, and both the small t's, if they get fertilized, it would result in a dwarf pea plant, which means 3 tall is to 1 dwarf, 3 is to 1. Phenotypically, it is 3 tall to 1 dwarf. But genotypically, observe the difference. Observe the genotypes of each of them. It is 1 capital T is to 2 capital T small t, because it's 2 of them, is to 1 small t small t. This 1 plus 2 together express the tallness. And that is what 1 plus 2 becomes 3 when it comes to the phenotype. Whereas 1 small t, even phenotypically, it is just 1, obviously, right? Because it's recessive. So this can be expressed in what we call the Punnett Square. That's the square that you see here. It's a tool that Mendel liked to use for these calculations because it appears neater and more systematic, right? So what you do is along the column, you express the gametes of one parent. Along the row, you express the gametes of the other parent. It can be the male or female, any way is fine. All you have to do is the cross multiplication of the two, because at the time of fertilization, sperm and ovum are coming together. If this sperm and this ovum together combined during fertilization, it results in this offspring. If this sperm fertilizes this ovum, it results in a heterozygote. Likewise, this and this together make a heterozygote, and the last two give you a homozygous recessive one. So obviously, what we were discussing, the phenotypic ratio is 3:1. 3 tall is to 1 dwarf, while the genotypic ratio is 1:2:1. Okay. This was Mendel's monohybrid cross. Likewise, he did it for all the other characters also, like for example, seed color, wherein yellow is said to be the dominant one and green is said to be the recessive one. So children, this is what I was referring to. As in, it's based on these observations that Mendel came to the conclusion that tallness is dominant over dwarfness. Okay, likewise, even in the yellow and green seeded experiments, Mendel, based on his observations, he came to the conclusion that yellow is dominant over green. So here, what do you think the parents would be? Parents would be purebreds, capital Y, and a small y. So you would get, in the F1 generation, you would get a heterozygote, and you end up self-fertilizing it, and what would you get? Look at it. So cutely shown, right, in this animation. Yes, you would get exactly the same results in the sense 1 capital Y, 2 capital Y, small y, and 1 small y, which means phenotypically it is 3 yellow seeded pea plants is to 1 green seeded pea plant. Okay, so this is about Mendel's monohybrid cross, and based on his observations in the monohybrid cross, Mendel came up with a couple of inheritance theories, and the first one is what we call the law of dominance, by which Mendel says that characters are controlled by discrete units called factors. Factors always occur in pairs, and in a dissimilar pair of factors, one member of the pair dominates the other. This is exactly what I told you sometime back also. This was Mendel's hypothesis. This is what he meant by law of dominance. Just remember any of the examples we have discussed until now, children. I think tallness and dwarfness is easiest for you to remember. And the law of segregation, which we also call law of purity of gametes, in the sense only one of the two gene copies present in an organism is distributed to each gamete which it makes, by which I told you, right? If it's capital T, gametes would have just one copy of the two alleles which are there in the parent. Whereas if it's a heterozygote, what do you think it is? They separate out. This would enter into one gamete and this would enter into another gamete. So basically, read it once again. The two gene copies get distributed to each gamete, which is the egg or the sperm cell that the organism makes, because the homologous chromosomes are what carry the pair of alleles or the genes, right? So during meiosis, in case you want to know more about meiosis, please watch my video on meiosis, which I did for ICSE class 10. That's already been done, I think a month or two ago. So please have a look at that. It will give you a better understanding. It will actually solidify your base in Mendelian inheritance. So yes, this is what happens, and during gamete formation, it ends up that a sperm carries only one copy of an allele. Okay, so this was the law of segregation, and because only one of the two copies enters into each gamete, we call it law of purity of gametes, because a gamete is said to be pure for a particular trait. Okay, the allocation of gene copies is random. It's randomly allocated, and when an egg and sperm join in fertilization, they form a new organism, and that organism's genotype would be a combination of the alleles which were contained in the gametes, in the sperm and the ovum respectively. Okay, so this is about law of segregation. Now let's move on to a step higher, and that is dihybrid cross, which is breeding between two plants differing in two pairs of contrasting traits. What do I mean by that? In a monohybrid cross, I told you, for example, in the tall and dwarf pea plants, I told you he chose pea plants which were similar in 6 characters but differed in the character plant height or stem length. Now in dihybrid cross, Mendel chose two parent plants such that they were similar in 5 characters, but in 2 particular characters, they exhibited contrasting traits. For example, he chose a pea plant which produced round and yellow seeds. Round seeds is one character, yellow seeds is another character. Seed shape and seed color, basically. And he also chose another parent which produces wrinkled and green seeds. Okay, so this would be the genotypic expression of these two, because they are also purebred. So obviously you can see that the main difference you see is that because it's a dihybrid experiment, there would be 4 alphabets that we use here. Okay, in the sense 4 letters we use, a combination of 2 alphabets. Basically, it can be 2 capital letters of one gene, 2 small letters of the other gene, or whatever. It can be anything, or it can be a mix of the two also. You will understand what I mean in some time. So the gametes would obviously contain only half of them, one copy of each of these genes, which means one allele for R and one allele for Y enters into one of the gametes. Likewise, in this case also, one R and one Y, because there has to be at least one copy representing each character's gene. Okay? And then what happens at the time of fertilization? In the F1 generation, it would be a double heterozygote, heterozygous for both of these. But then phenotypically, physically, Mendel observed that they expressed only the dominant traits. Okay, round yellow. Wrinkled green was nowhere to be seen. So what did Mendel do? Mendel went about self-fertilizing them, just as he did for monohybrid cross. So again, Punnett Square is certainly the best way of working out for a dihybrid cross. So let's write the gametic combinations here, children. For one of the parents, as in for the sperms, the different sperm types would have gametic combinations of capital Y, capital R, capital Y, small r, capital R, and a small y, small r. Likewise, for the other parent also, it's a capital Y, capital R, capital Y, small r, small y, capital R, and a small y, small r. Okay? So, yeah, so this would cross multiplying both in the sense, if this particular sperm fertilized this particular egg, the resulting offspring would have this as its genotype. If this sperm and this egg combine together, it would have this as its genotype. I will just work out one row for the time being, okay, because it will take some time. And this case would be a capital Y, small y, capital R and a small r. Now children, do remember whether you write the small y first or the capital Y first, or the small r first or the capital R first, it doesn't really matter, because just like in mathematics where you write 2 into 3 is 6 or 3 into 2 is 6, both are giving you the same result. Okay, but then the only thing to take care of is that it's always better to write the two y's together and the two r's together. It will be very confusing to write yr. Okay, alleles of one gene should ideally be written together. That's the correct way of writing it. And gametes should ideally always be written with a circle around it. That's one correct way of expressing these. Okay, so what do you get? Finally, when you fill up all the 16 boxes, this is what you would see. You can see that it's a mix of so many different combinations. And what exactly are they? Yes, you would see that there are 9 yellow round. What are they? This one, this 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 yellow round. Okay, wherever there is one capital Y and one capital R, at least one capital Y and one capital R, it would be a yellow round phenotypically. Okay, and next is yellow wrinkled. There would be 3 of them which are yellow wrinkled, by which I mean one capital Y, at least one capital Y, but both small r's, because that is what gives the wrinkled appearance. So 1, 2, and it would be 3. Okay, 3 yellow wrinkled, and then it would be 3 green round. Okay, green round would be these 3. Green because both small y's homozygous for that, and at least one capital R would make it round seeded, and 1 green wrinkled, which makes the phenotypic ratio 9:3:3:1. Now, what about the genotypic ratio? Genotypic ratio, you will have to sit and work out actually. You would see that it's a complex ratio, as you can see, 1:2:1:2:4:2:1:1. Now, do remember, genotypic ratio is commonly not asked in exams. In dihybrid ratio, dihybrid crosses, genotypic for monohybrid is commonly asked, but for dihybrid, commonly it is only the phenotypic ratio which is asked. But it's always worth knowing, so I would leave it to you to sit and figure out what exactly 1 is, what is 2, what is 1, 2, 4, 2, 1, 2, and 1. For example, there is only one genotype as capital Y, and a capital R, capital R. There are 2 genotypes which are capital Y, and capital R, small r. And you would find 4 genotypes which are double heterozygous, and so on. That is what gives you the genotypic ratio like this. Okay, so this is what Mendel observed in his dihybrid cross, and based on this, he came up with the dihybrid law of inheritance, which is called law of independent assortment. And according to this, the alleles of 2 or more different genes get sorted into gametes independent of one another. Okay, so what happens is, like in simple words, I can tell you, the allele which a gamete receives for one gene does not influence the allele received for another gene. Okay, allele received for one gene does not influence the allele received for another gene. For example, when I say a gamete has received the capital Y allele for the seed color gene, it doesn't mean that for the other character, which is seed shape, it doesn't mean that for the other one also it has to be a capital R. It can also be a combination of capital Y, small r, or it can be a combination of small y, capital R. It can be a small y, small r, and so on. Okay, so both of them are independently assorted. The allele a gamete receives for one gene does not influence the allele received for another gene. This is what the law of independent assortment states. Okay, so let's just look at Mendel's laws at a glance. These are the 3 major laws: the law of dominance, the law of segregation or law of purity of gametes, and the law of independent assortment. What is the significance of this? Mendel's laws help us even today because desired qualities can be improved to improve plant quality as far as agriculturists and plant breeders are concerned. That's one major advantage. Secondly, it helps to predict phenotype and genotype of offsprings. Now, for example, even in my case, because my husband and I are both O positive blood group, because of Mendel's laws, and because I have studied genetics in my school and college days, I obviously I know how blood group inheritance occurs. So obviously I knew that whether I have one child, two children, three children, or whatever it is, all of them are going to be of O blood group. So children who are curious to know blood group inheritance, probably like I will make a separate video on explaining how blood groups get inherited. But I think for ICSE, I have already made videos on that blood group inheritance. In case you're interested, you can go ahead and watch that. And thirdly, it makes understanding of complex patterns of inheritance very simple. It's no rocket science. It's not any rocket science. As I said, even common people, as long as you know the basic rules, you will be able to predict and you will be able to understand it logically. It's not just about pea plants. Whatever Mendel came up with is applicable to every sexually reproducing organism. So let's now answer the last few questions for today's session. The first one being the F2 genotypic ratio of a monohybrid cross is dash. F2 genotypic ratio. How is it commonly expressed? And the answer is 1:2:1. 1 capital T. Let's recall. 1 TT is to 2 heterozygotes plus 1 small t. F2 genotypic ratio of a monohybrid cross. Next one. Which of these is also known as the law of purity of gametes? Okay. Is it the law of dominance, law of segregation, or the law of independent assortment, or both B and C? And your time is up. The answer is law of segregation. We say that a gamete is pure for a particular trait. The next one, dash is the F2 phenotypic ratio of a dihybrid cross. F2 phenotypic ratio of a dihybrid cross. And your time is up. The answer is 9:3:3:1. Those of you who are still confused, go back to the dihybrid Punnett Square, try counting it for yourself and see if this is coming correct. The next one. In the genotypic ratio 1:2:1 of the monohybrid cross, how many are true recessive? How many are true recessive? 1, 2, 3, or 4? Your time is up. The answer is 1. As I said, it is 1 capital T to 2 capital T small t is to 1 small t small t. True recessive is this. It's only 1 in this ratio. Okay, the next one. It's a general question which I have not told you today, but you should be able to answer this from your common sense. Removal of stamen from the flower is termed as emasculation, pollination, breeding, or none of these. Now, why am I asking this in the context of genetics is because Mendel followed this particular step to ensure that pea plants did not self-pollinate. In order to allow cross-pollination to happen, in order to be able to perform cross-pollination, Mendel performed this particular step. What was it? And your time is up. It is emasculation because stamens are the masculine part or the male part of a flower. So emasculation is removal of stamen from a flower. Okay. And one more last question for you. The F2 phenotypic ratio of Mendel's monohybrid cross is dash. 3:1, 1:3, 1:2:1, 2:1:1. And your time is up. The answer is 3:1. F2 phenotypic ratio. And what exactly does that stand for? 3 tall is to 1 dwarf. In the case of plant height or stem length, this is the case. Otherwise, it's in the case of seed color, it would be 3 yellow is to 1 green. Or if it's pod color, it would be 3 green is to 1 yellow, and so on. So that's about it. Great job, all of you who have got everything correctly, everything correct. Rest of you, not to worry. I would suggest it's very, very important to have a proper base, proper understanding of this chapter so that, honestly speaking, this is one of those chapters which needs minimal memorizing. Okay. Unfortunately, ICSE children have this chapter removed from their syllabus. Unfortunately for me and unfortunately for some of them. So if there are any doubts that you want me to answer, children, please do put it in the comment section below, because as you know, at the end of every chapter, we have a live doubts plus mentee session. So we will be taking up the most commonly asked doubts from the comment section below of each of these sessions, and we will be answering them. So let me remind you that you now know about Mendel and the pea plant, what is monohybrid cross, what's dihybrid cross, the 3 laws of Mendel. And yes, here is your homework question. The factors mentioned by Mendel are now known as genes. Where are these genes found? Where are these genes found? We've spoken a lot about them. Where do you think you would find them? Give me the answer to that, and let's see who's coming up with the fastest answer. And do remember, children, to visit the link given in the description box below and use the coupon code AMBPRO to avail the best benefits. And of course, if you have enjoyed this session, if you have found it useful, please, please, please do not forget to hit the like button right now. Please do share it with all your friends of CBSE Class 10 also, and even your NEET aspirant friends, because this will benefit all of you guys. And please do subscribe to our channel Vedantu 9th and 10th English in case you haven't done it yet, because this is not just a one-time thing. We aim at covering your entire syllabus so that all of you benefit even in this time of online schooling and things like that. Everyone has to benefit from this. Ensure we are also here to ensure that your learning never ends. And yes, of course, after your syllabus is done with, we will be coming up with more and more creative sessions for your revision and things like that. So let's think about it in a couple of months from now. Until then, do remember to take care, stay happy, and stay healthy. This is Ambika signing off. Bye bye.